This is our last video, and it's a short one. We're just going to talk about the mechanism of action of hormones. So, how hormones act depends on whether they are hydrophobic or hydrophilic, and that is because, as seen in this picture, if they are hydrophobic, like these steroid hormones, they will be linked to a carrier protein and they will have to be removed from that carrier protein to pass through the lipid bilayer. And the receptor for these hydrophobic hormones are going to be intracellular or even on the nucleus. So here we have steroid hormone specifically. It is bound to a carrier protein, but it has to be removed from that carrier protein to go into the cell by going through that hydrophobic phospholipid bilayer. And inside the cell, we will have the receptor protein for this steroid hormone. Then what happens is this receptor protein with the hormone bound is going to translocate, which means it's going to move inward and go and attach itself to DNA. And what that will do is that is going to change the process of transcription and translation, which is going to impact protein synthesis, which means the steroid is acting on protein production. So it does this by attaching on to its receptor protein, and then the receptor proteins attach onto our DNA. What actually happens for these steroid hormones specifically is that there are two sites for which receptor proteins attach. And so they're called half sites, and two different receptor proteins with hormones attached bind and they join together. And so this is called dimerization. They bind to that DNA, and that is going to impact the synthesis of RNA and then impact protein synthesis. So this is called genomic action, and it is slow. It takes at least 30 minutes. And so this just goes to show how slow acting hormone effect can be. So we've just outlined how steroid hormones, which are hydrophobic, work, but what about hormones that are hydrophilic? So these would be like hormones like epinephrine or norepinephrine, ADH. So some of our amines, our polypeptides and proteins, and our glycoproteins could possibly be hydrophilic. And because these are hydrophilic, they cannot pass through that phospholipid bilayer of the plasma membrane. And so they will have to have surface receptors on the cell. And by the way, a big reminder that these hormones are only going to attach to cells if they have the receptors for those hormones, and not every single cell is going to have this. And so these water-soluble hormones are going to attach via those cell surface receptors, but to instigate change within the cell, those are going to be mediated by second messengers. The hormone is the first messenger, and then it's going to cause like a cascade effect inside the cell using a second messenger. So there's three main types of second messenger systems used by these water-soluble hormones, and we're going to talk about each type. So first off, we have adenylate cyclase and cyclic AMP. And so here, we've seen this before, we have a hormone, a hydrophilic hormone, attaching to a cell surface receptor. And that cell surface receptor is also attached to a G protein. G protein subunit. So alpha, beta, gamma, G protein. And that's going to cause this subunit to dissociate, and that's going to activate an enzyme called adenylate cyclase. And this is going to convert ATP to cyclic AMP. And we've seen this before. Cyclic AMP is a second messenger. That is our second messenger. It is going to activate protein kinase, which is going to phosphorylate protein and activate it. So that is how an enzyme gets activated by that first messenger of hormone. Next, we have a system called the phospholipase C calcium system. So again, we have that hormone attaching to receptor protein with the G protein subunits going to dissociate and then activate phospholipase C, PLC. And that is going to form a compound called inositol triphosphate, IP3. And IP3 causes calcium channels to open in the endoplasmic reticulum, and calcium here is our second messenger. Calcium is going to bind to calmodulin, and this is going to activate enzymes within this cell. And last, we have a tyrosine kinase, and we actually have seen this before too with insulin. I showed you this picture in the last video of how insulin is bound to these cells, and it's going to cause translocation of these vesicles. It's going to cause these vesicles to come to the surface so that glucose can be taken in via these cells. But these use a tyrosine kinase system. So the receptor of the insulin receptor itself is an enzyme, tyrosine kinase, and notice here that the insulin binds to 2 units of the receptor, and that is going to allow for these receptors to come together, and aka dimerize, and the enzyme tyrosine kinase gets activated. It adds a phosphate group to itself, and this is what allows for the cascade of effect and for glucose to come into these cells.